Welcome to Biochemistry 2. A Lecture 0 has been uploaded to OneDrive. Please look through this lecture for additional information regarding the course. Think of it as an extension to the syllabus. This is the first lecture of a two-part series that covers the very fundamentals of Biochemistry 1. Biochemistry 2 will concentrate on structure and function of protein complexes involved in lipid biosynthesis, amino acid oxidation, amino acid biosynthesis, DNA metabolism, RNA metabolism, and protein metabolism. The slide number in this lecture are not in order because they originate from multiple Biochemistry 1 lectures. Don't freak out because the numbers at the bottom right are not continuous. Amino acids, peptides, and proteins. A partial review of Biochemistry 1. During the lecture, I will be asking you questions. Please think about these questions and come up with an answer on your own. Feel free to ask these questions during the Blackboard Collaborate Ultra session of the course. We will begin each presentation with an outline of what we will be discussing in the presentation. The 20 amino acids can be categorized into different groups based on the chemical property of their side chains. The first group discussed is called the nonpolar group. The side chain is not able to hydrogen bond or make electrostatic interactions with other chemical moieties. These amino acids are predominantly in the hydrophobic core of a cytosolic protein. Where would you anticipate these amino acids to be for transmembrane proteins? Glycine and alanine can be on the surface of cytosolic proteins because of their small size. Why does size matter? Proline side chains form a covalent bond with the main chain amine. What are some of the properties of proline that you remember from Biochemistry 1? There is also the polar group. With the exception of one amino acid, the side chains of these amino acids are composed of atoms with slightly different electronegativity than carbon. This increases the solubility of these amino acids. Consequently, these amino acids are predominantly found on the surface of cytosolic proteins, where they can form hydrogen bonds with the solvent. They can also be found in the interior of proteins. However, what requirement must be met for this to happen? Two properly positioned cysteines can form a disulfide bond. What two roles do disulfide bonds play? Aromatic amino acids are conjugated. This property allows them to absorb light. How is the complexity of a conjugated system related to the wavelength of light the molecule absorbs? Think back to your organic chemistry days. These amino acids are also hydrophobic and thus not very water soluble. They tend to be in the hydrophobic core of proteins. Their ability to absorb light can be used to determine the concentration of protein in solution. Please brush up on the Lambert-Beer law you learned in general chemistry if you don't understand what I'm talking about. What assumption must you make in order to use the Lambert-Beer law to estimate the concentration of a protein? These are the basic and acidic amino acids. These amino acids can be protonated and deprotonated depending on the pH of the solution containing the protein. Do all aspartic acids in the polypeptide possess the same pKa value? Why or why not? Here we review how the main chain amine and carboxylic acid groups of two amino acids condense to form a dipeptide. The bond highlighted in yellow is an amide bond. Can you remember two properties of the amide bond? This condensation reaction is primarily done by the ribosome during translation. We will discuss this process during the latter part of the course. Here is a hexapeptide. You should refamiliarize yourself with the structures and properties of amino acids. Structure or protein structure is extremely important in this course, so you need to know what amino acids look like. You can estimate the molecular mass of a peptide or polypeptide by multiplying the number of amino acids by 110 Daltons. What is a Dalton? Proteins can be quite large. Do peptides or polypeptides always have an N-terminus and a C-terminus? Proteins can be thought of as long peptides, or a string of amino acids. Some strings of amino acids automatically fold by themselves to form proteins. Some proteins can be denatured, unfolded, and will refold to the native structure by themselves if placed in the proper buffer. Other proteins need the help of molecular machines or other protein complexes for refolding. There are 4 levels of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure refers to the amino acid sequence of the polypeptide. Secondary structure refers to an alpha helix, a beta strand, and a gamma or beta turn. Can you name 2 properties of an alpha helix? Other than the structure, which is the geometry of the backbone, what is the biggest difference between an alpha helix and a beta strand? Tertiary structure is composed of several secondary structures. These secondary structures can be purely helical, purely beta strands, or a mixture of the two. Quaternary structure refers to 2 tertiary structures interacting with one another. Do you remember the first quaternary structure you saw in Biochemistry 1? Proteins can be modified with prosthetic groups. These are non-amino acid molecules that become part of the protein. This process occurs post-translationally, meaning after the protein has been synthesized by the ribosome. There are enzymes that are responsible for identifying the target, which is an amino acid on a protein, and the ligand, and putting the two together. These modifications may be, but need not be, covalent modification. How can a protein be covalently modified? Examples of some prosthetics groups include lipids, carbohydrates, phosphates, hemes, flavonucleotides, and metals. Amino acids can also be modified with amino acids. What example of this have you seen in this lecture? I'm now going to present some slides that you may have not seen before, depending on who your instructor was for Biochemistry 1. The reason I'm presenting these slides is because it's important that you become familiar with how proteins are attained. This is important so that you have a better understanding of what's happening in the manuscripts that you will be reading and you will be providing reports and presentations on. Cellular fractionation. Cellular fractionation is one of the earliest techniques used to purify proteins. An organ or tissue served as the source of proteins. Organs can be broken using a blender. Cells can be broken using a homogenizer, a sonicator, or a French press. The idea behind all of these techniques is to break the cell to release its contents. Obviously, you would need the appropriate buffer in both situations to extract the protein of interest. Remember, proteins are fragile, and some can be very particular with what buffer they can tolerate. So, identifying the proper buffer to purify the protein of interest can take some time to identify. We will not discuss this too much in this course. The solubilized material is referred to as the lysate. The cytosol, organelles, and the membrane. Breaking the cells requires breaking the membranes that surround the cell. Remember that membranes are not water-soluble, so you need to separate them from the water-soluble components of the cell. Organelles are also released when eukaryotic cells are broken. The easiest way to separate the membranes and organelles from the water-soluble components is centrifugation. Two types of centrifugation are used to purify the components of the cell: differential centrifugation and isopycnic centrifugation. Differential centrifugation. In this technique, the lysate is centrifuged at different speeds to separate the cellular components in the buffer. The speed at which the rotor turns and the size of the rotor determine the amount of force experienced by the material inside the test tube. Given that the contents inside of the test tube have different molecular weights, they will settle to the bottom of the test tube under different centripetal force. The speed and time are empirically determined. In this technique, you are separating material based on their molecular weight. The heavier material will settle to the bottom of the tube under lower centripetal force. Differential centrifugation is always performed before isopycnic centrifugation because the first step of purification requires the separation of cytosolic components from membrane components. Isopycnic centrifugation. In this technique, the lysate is carefully layered on top of a solute gradient. In this slide, it's a sucrose gradient. What this means is that you have a solution of high sucrose concentration at the bottom of the test tube, which is a more dense solution, and a low sucrose concentration at the top of the test tube, which is a lower dense solution. The test tube is then centrifuged at a particular speed for a particular amount of time. The speed, time, and concentrations of sucrose are empirically determined. Material will separate according to their density and will stop moving down the tube once they locate to a region with a density that is similar to their own. In this technique, you are separating material based on their density. This is also a crude technique because many different biological materials possess the same density. When setting up this density gradient of sucrose, why is it that we use high concentration of sucrose on the bottom and a low concentration of sucrose on top? Meaning, why don't we use a low concentration of sucrose on the bottom and a high concentration of sucrose on top? Is there something wrong with this idea, or is it perfectly fine? Generally, multiple techniques will be combined to separate material according to multiple properties. In this slide, we combine differential and isopycnic centrifugation to separate material in two dimensions: molecular weight and molecular density. Some material can be separated using only two of their properties. Other material may require the use of multiple molecular properties for purification. What molecular properties can you think of that can be used for purification? Protein purification. Some more recent techniques involve chromatography. The main advantage of this technique is that you can process larger volumes. The second advantage of this technique is that you can separate material based on additional properties. These techniques can be combined with the above two techniques to separate material based on several properties. Affinity chromatography. In affinity chromatography, you take advantage of the specific affinity between your protein and a ligand to purify the protein. For example, you can genetically modify your protein to possess an amino acid sequence that has high specificity and affinity to a particular ligand. You can synthesize or purchase chromatography resin that is covalently attached to this ligand. You then cast a chromatography column that has this resin inside. This setup looks very similar to the silica columns you ran in your organic chemistry labs. By running your lysate through the column, you will allow your protein of interest to bind to the ligand, which is attached to the resin. Thus, your protein of interest is immobilized on the resin. You then run buffer through the column to wash all proteins that are not bound to the resin. These are proteins that have no affinity to the ligand or the resin. You then elute your protein by including a high concentration of the ligand in the buffer. In this scenario, the protein is binding and releasing the ligand in your buffer as well as the ligand attached to the resin. However, because the concentration of ligand in solution is greater than that attached to the resin, the protein will predominantly interact with the ligand in buffer and elute from the column. Make sure you understand this concept. You have seen this phenomenon throughout Biochemistry 1 and will continue to see it throughout Biochemistry 2. The phenomenon dates back to your general chemistry studies. Go back and review your general chemistry notes if need be. With this technique, you can separate based on the binding partner of a molecule. Ion exchange chromatography. Charge is used in ion exchange chromatography to attract or repel proteins. You can use either positive or negative charged chromatography resin. Thus, if your protein has lots of positive charge, it will stick to negative charged resin, and vice versa. Why would a protein have a net positive charge? Why would a protein have a net negative charge? Most proteins will carry some charge, and most proteins will bind to cation or anion resin. Cation chromatography will bind to proteins with a net positive charge. What charge does this resin have? Anion exchange chromatography will bind to proteins with a net negative charge. What charge does the resin have? Because proteins or molecules of interest carry different net charges, their affinity to the resin will be different. Thus, some materials bind to the resin with higher affinity and others with lower affinity. To release the material from the chromatography resin, one can use sodium chloride to compete with the resin. This is because sodium has a positive charge and chloride has a negative charge. Thus, a gradient of increasing sodium chloride concentration is processed through the column to separate material of increasing affinity for the resin. Material with the lowest affinity for the charged resin leave the column first, and material with the highest affinity leave the column last. With this technique, you can separate based on the charge of the molecule. Imagine two proteins with identical positive net charge, but one protein binds to the chromatography resin with higher affinity. Come up with two reasons why this would happen. Size exclusion chromatography. This technique separates material based on their size. The chromatography resin has many tunnels built into it. These tunnels have various radii. For simplicity, imagine that a single tunnel has a single radius. Molecules that are smaller than the tunnels can pass through the tunnels, whereas molecules that are larger are excluded from the tunnels. The length and number of tunnels increase as their radii decreases. This means that smaller material can travel longer distances. The consequence of this is that larger material have less and shorter tunnels to travel through. Thus, they elute earlier because they have a shorter distance to travel through. Smaller materials elute later because they have more and longer tunnels to travel through. With this technique, you can separate based on the size of the molecule. Protein purification. Here is a potential strategy to purify a protein from cells. Read through this and think about what is happening. Feel free to ask specific questions during the lecture. SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis, is the most used technique in the field of biochemistry. It is absolutely impossible to purify a protein without having to use SDS page. SDS page allows the user to separate proteins based on the length of the polypeptides. Thus, this technique allows the user to determine the molecular weight of the polypeptide. As you read through the slide, I want you to ask yourself, what does SDS look like? Where have I seen something like this before? Again, refer back to the courses you took before this course. What is the major disadvantage of this technique? Here is a slide describing how SDS page is run. Samples are loaded into the channels at the top of the page. Current is passed through the page, and polypeptides are separated based on their size. Larger polypeptides are at the top, and smaller polypeptides are at the bottom. The physics behind this process is not important for this course. However, it may be important for your biophysics course. This is an example of what an SDS page looks like. The blue bands identify different proteins. You can use this technique to identify the purity of your protein and the molecular weight of your sample. Tell me the major problem with this technique. This is an extremely important question because there is a limitation with every technique used in science. Isoelectric points of proteins. The isoelectric point, or pi for short, of an amino acid is the pH at which the amino acid carries a net charge of 0. You need to use the pKa of amino acids to calculate their pi, and you should remember how to do this for each amino acid. The same exercise is carried out for a protein. However, there is a difference between the pKa values of protein amino acids and isolated amino acids. Do you remember what and why this is? Proteins can be separated according to their isoelectric points. Isoelectric focusing. In this technique, an electric field is used to isolate proteins according to their charge. To do this, a pH gradient is established in a test tube. In this example, we have a pH of 9 at the top of the test tube and a pH of 3 at the bottom of the test tube. The protein is placed at the very top, and an electric field is applied to force the protein through the gradient. The protein stops moving when it reaches the pH that is equal to its pi. This is because the protein has a net charge of 0 at this pH. Within the cell are thousands of proteins, and many of these proteins have the same pi. So pi alone is not sufficient to isolate or identify a protein. However, it is a very good start. By combining isoelectric focusing with SDS page, one can separate proteins based on their pi and size. This is referred to as a two-dimensional gel because there are two dimensions, pi and molecular weight. This technique allows one to observe the change in charge of proteins within the cell. More sophisticated equipment can extract and properly identify the band that has shifted to give the identity of the protein that has been phosphorylated or modified. Here is an image of what a two-dimensional gel looks like. As you can see at the top is the isoelectric focusing, and on the right-hand side is the SDS page. Each spot is identifying at least one protein. The cell, under different circumstances, will provide a different profile, and these profiles can be used to learn more about the cell. This is something we're not going to get into for this course, but I think it's important for you guys to realize that you can combine, in fact, in many instances, you combine multiple techniques to give you a better understanding of what is happening. These next two slides provide a more quantitative means by studying the contents of cells. You can use protein sequencing to identify the amino acid sequence. This is something you guys saw in Biochemistry 1, and you're seeing again here. These are extremely important techniques that we really can't do without. I'm not going to go into the details of these things, of these techniques, but I do want you to be familiar with them. So take some time, read the slide, and ask questions if you have any.